Hello, my name is Dr. Jessica Hua. I'm very excited to have this opportunity to be a part of this conference. I'm looking forward to sharing a little bit more about what my lab is doing at Binghamton University. Okay, so today's talk will focus on disease ecology in the face of pollutants. So to begin broadly, we all recognize that amphibians are facing a wide range of different challenges, from emerging infectious diseases, changes in our climate, pollutants in our environment, and also habitats that are being lost or modified in unique ways. And so under this broader umbrella of global change biology, my lab and today's talk will focus on understanding how global change influences host-parasite interactions in amphibians. So when it comes to understanding host-parasite interactions, one approach might be to ask, well, what are factors that influence a host's ability to defend itself against parasites? Alternatively, from the parasite perspective, we can ask, what are factors that influence the parasite's ability to infect hosts? So building upon this traditional model, the field of disease ecology has increasingly recognized the importance of the environment in shaping disease outcome. Me being trained as an ecotoxicologist, in my lab, we're interested in how pollutants in the environment, from things like pharmaceuticals to light pollution, how pollutants can influence this interaction between hosts and their parasites. Now, while this disease triangle framework provides a really nice foundation for understanding how pollutants influence hosts and their parasites, one limitation is that we recognize that in nature, hosts and parasites, their interactions don't occur in a vacuum. For instance, there are often multiple host species interacting with each other, and this may affect the disease outcome. Similarly, there might be other stressors in the environment, like invasive species that might interact with pollutants to then affect disease outcome. And so a central theme in my lab and for this talk is to ask, how can we integrate ecological complexity into our understanding of disease outcome? Towards this goal, the first theme of today's talk is an ecological perspective. I'll be talking to you about a tiered approach that we're taking, and we'll be telling you about some research on secondary salinization of amphibian habitats. Specifically, we'll be asking three questions. First, we'll be asking, how does sodium chloride influence simple host-parasite interactions, so single host, single parasite interactions? From there, I'll start layering on a little bit more complexity by asking, how does salt influence host-parasite interactions in multi-host communities? And then lastly, at the ecosystem level, I'll be asking, how do changes in abiotic conditions interact with salt to affect hosts and also their susceptibility to parasites? So in addition to this theme of integrating ecological complexity, we know that global change biology is a topic that is intimately linked with human society. And so it's important to me and also members of my lab that we find ways of more effectively communicating our science to broader audiences. And so in addition to the ecological research, in this section, I'll be telling you all about some of our research in science communication and outreach, specifically how we have used art to facilitate science communication when it comes to our community science initiatives. And for reference, this portion of the talk will fall in between or right after the multi-host portion of the ecology section. So this is the outline for today, and I'm going to jump right into talking about this research. So first, secondary salinization is the influx of salt water, so NaCl, into freshwater ecosystems. There are a variety of ways that this can occur, from coastal flooding, where seawater comes into amphibian habitats, freshwater amphibian habitats, to the use of road salts to protect roadways from icy conditions, to also agricultural practices such as irrigation, which can bring salts into the fields, and these salts can then run off into freshwater systems like ponds. So to understand how secondary salinization influences host-parasite interactions, in my lab, we focus on three different types of parasites, amphibian parasites: ranavirus, or Fe3, trematodes, or chytrid. For the sake of time, today I'm going to focus this talk on trematodes. What you're seeing on the screen now, this is the free swimming stage of the trematode, the cercaria. This cercaria form will infect larval amphibians by swimming up the cloaca of the tadpole and encysting in the kidney tissue. So the image right below the tadpole there, these are metacercarial cysts in the kidney of the tadpole. So to understand how sodium chloride modifies host-parasite interactions, I'll be focusing today on wood frogs, Rana sylvatica, and the model parasite will be trematodes in the free-swimming stage, Echinostoma trivolvus, the cercaria stage. To understand how sodium chloride modifies host-parasite interactions, I'll be telling you first about some work from my former PhD student, Nicholas Buss. He took wood frog tadpoles and exposed them to 4 different concentrations of salt. For reference, seawater is about 19.4 grams per liter of chloride, The highest concentration of chloride that was detected in freshwater was from runoff. It was about 18 grams per liter. The highest concentration detected in a wetland was about 4.6 grams per liter, though the majority of wetlands have chloride concentrations ranging anywhere from zero point 0.1 to 1.1 grams per liter. So these are concentrations. First, they are environmentally relevant concentrations, including one that falls within the range that is commonly found in wetlands. And then second, these concentrations are sublethal to our wood frogs. The lethal concentration for wood frogs is just above 4 grams per liter. So after exposing wood frog tadpoles to these 4 different concentrations, Nick exposed them to 50 of the free-swimming trematodes. He then counted how many of these 50 trematodes successfully encysted in the kidney of the tadpole. In this slide, on the x-axis, we have sodium chloride concentration. On the y-axis, we have the average number of cysts. When not exposed to salt, we found in this white bar here, on average, about a little above 12 cysts per individual. In comparison, when exposed to sublethal concentrations of salt, we found that in all cases, exposure to sublethal concentrations of sodium chloride increased tadpole susceptibility to trematodes. So taking a step back, what are the main take-homes here? We found that sublethal concentrations of salt can modify host-parasite interactions, but what I think the main take home is that in the field of toxicology, we tend to focus on what are the lethal concentrations of a pollutant. We think that studies like these underscore the importance of also considering environmentally relevant and lower concentrations of pollutants because while they may not have direct lethal effects, they may still harm amphibians by influencing their interactions with enemies like their parasites. Now, of course, one limitation of this study, as I alluded to in the beginning, is that we focus on a single host and a single parasite. In reality, in nature, you often have multiple hosts of amphibian interacting with each other, as well as with their parasite. In fact, this relationship between host biodiversity and disease is a topic that has been hotly debated in the field of disease ecology for the past 2 decades. This idea of the biodiversity disease risk. So what is the biodiversity disease relationship and why are people so interested in it? And when it comes to the biodiversity disease relationship, one pattern that commonly emerges is the effect called the dilution effect. This is an effect where as species diversity increases, you get a decrease in overall infection. And so this effect has a clear benefit for why. So this dilution effect relationship. So this relationship between biodiversity and so why people are so interested in this relationship is that it provides a clear benefit to why we might want to protect species diversity. By protecting diversity, we get the benefit of reducing overall infection in that community. The challenge, however, is that while we do see the dilution effect many times in nature and many times in experimental studies, sometimes and oftentimes we find no relationship, or alternatively, the opposite relationship, amplification, where increased species diversity leads to increased overall infection. And so in this section of the talk, what I'd like to highlight is one factor that we think might be contributing to the equivocal nature of this relationship between species diversity and overall infection. Before I do this, I wanted to spend one slide to talk a little bit more about what are the mechanisms that might be driving the dilution effect. To do this, I've generated 2 hypothetical communities. The community on the left is a lower host diversity community with just one species, the blue tadpole. The community to your right is a community that has higher host diversity with 3 species of tadpole, blue, red, and yellow. So drawing your attention to the lower host diversity community, we can imagine that these hosts are in this scenario are highly susceptible to the parasite. So with parasites in the water, these highly susceptible hosts are very likely to draw the parasite and become infected. So in this scenario, you get 100% infection in the lower host diversity community. In contrast, in the higher host diversity community, theory predicts that we're more likely to have hosts that are less susceptible to the parasite. And these less susceptible hosts can serve as decoys in the community. In other words, they draw parasites to them, but because they're less susceptible, these parasites don't successfully infect the host. And so what we see is that in the presence of higher host diversity, we see a benefit of reduced overall infection. By having these decoy hosts in the community, these decoy hosts can protect the rest of the community from infection. Graphically, it would look something like this. As diversity increases, you see a decrease in overall infection. This is the dilution effect. One challenge with these types of studies is that they often occur in benign environments. This is relevant because, as I just showed in the previous study, the presence of a pollutant like sodium chloride can change patterns of susceptibility. So looking back at our higher host diversity community, in the presence of salt, these less susceptible hosts may no longer be less susceptible. Salt can increase susceptibility to parasites. in this situation, we see that despite having higher host diversity, these communities no longer have reduced infection because the less susceptible host became more susceptible. And so it may look something like this on the figure. In the presence of salt, we no longer see that dilution effect. In the absence of salt, we do see the dilution effect. And so taking a step back, we predicted that pollutants can influence the magnitude of the biodiversity disease relationship. To test this prediction, we had to ask two questions. First, how does sodium chloride modify host susceptibility to parasites? And then once we understand how salt changes patterns of susceptibility to parasites, we can then make predictions about whether salt can modify the magnitude of this biodiversity disease relationship. Again, this is work done by my former PhD student, Dr. Nicholas Buss. So how does salt modify host susceptibility to parasites? What Nick did was he repeated that first study, except this time with different species of amphibian. So he took peepers, wood frogs, and toads, exposed them to no salt or 1 gram per liter of sodium chloride, then exposed them to 50 trematodes and counted how many successfully encysted. On this data slide, on the x-axis, we have the 3 different species. On the y-axis, the average number of parasites per community. What I'm going to show first are the species in the absence of salt. What you can see is that when salt is not in the water, peepers, wood frogs, and toads were all similarly susceptible to the parasite. In contrast, in the presence of salt, we found that peepers became 34% more susceptible to parasites. Wood frogs became 64% more susceptible to parasites, so similar to that original study. And then for toads, we found no difference. Toads were similarly susceptible in the presence or the absence of salt. So now that we understand how salt can change patterns of host susceptibility to parasites, we can then make predictions about whether salt contamination can modify this relationship between species diversity and infection. So starting in a community in the absence of salt, we found that peepers, wood frogs, and toads all have very similar susceptibilities to the parasite. So in this scenario, there's no one species that can act as a decoy to draw parasites away to reduce overall infection. So we see that we would predict that there's no benefit to increasing species diversity. In the absence of salt, there's no diluter species. Increased species diversity, we would not expect to find a dilution effect. In contrast, in the presence of salt, what we find is that toads are significantly more tolerant to the parasite compared to both peepers and wood frogs. And so what we predict is that as species diversity increases, specifically as species diversity increases with any community that includes a toad, we would see the toad acting as a decoy, causing a decrease in overall infection. So we would predict that in the presence of salt, we would see a dilution effect. So to test these predictions, what we did was we generated 7 different communities, 3 communities that only had a single species, 3 communities that had 2 species, the combination of the single species, and then 1 community that had all 3 species. One thing to point out, while we manipulated the number of species in the community, we kept the number of tadpoles consistent at 6. After generating these communities, we reared them in either salt or no salt environments, added trematodes, and then counted how many successfully encysted. On this data slide, I'll be showing results from the communities that were not exposed to salt. To orient everyone to the x-axis, from the left to the right, we go from communities of lower host diversity to higher host diversity. On the y-axis is the CIS per community. For the first panel, we're comparing all the communities back to single peeper communities, the middle panel all back to single wood frog communities, and then the last panel all back to single toad communities. As a reminder, we found that peepers, wood frogs, and toads all had similar levels of susceptibility, so we didn't predict that increasing host diversity, we would not see that benefit of reduced susceptibility. So we predicted no evidence of a dilution effect. So what did we find? We found that in all cases, we found no evidence of a dilution effect. Increasing host diversity did not lead to a decrease in overall infection in the absence of salt. So in the absence of salt, no dilution effect. In contrast, as a reminder, in the presence of salt, what we found was that toads were significantly more tolerant to parasites compared to peepers and wood frogs. And so because of this shift in patterns of host susceptibility, we can then look to how this influenced that biodiversity disease relationship. On the x-axis here, we have the communities. On the y-axis, we have the number of cysts per community. Peepers and wood frogs were highly susceptible to the parasites in the presence of salt. Toads were less susceptible. So we predicted that in any community that included a toad, we would see a decrease in overall infection. For the community that had the most species, the 3 species community, we predicted that there would also be a reduction in infection. But because in this community, there are only 2 diluter species, the toad, to 4 susceptible species, we predicted that the decrease would not be as high in magnitude. So what did we find? Here are the average number of cysts per community of just peepers compared to peeper plus toad communities, and we find a significant decrease in infection by 28%. Compared to wood frog only communities, when you have wood frogs plus toads, we found a significant decrease by 45%. And then lastly, in the most diverse community, we find that there was a significant decrease compared to peeper only communities by 30% and a no change compared to wood frog only community. So put all together, what did we find? We found that sodium chloride exposure altered host susceptibility to parasites. By changing patterns of host susceptibility, we found that this modified the biodiversity disease relationship. And so to some, we found that salt can change patterns of susceptibility, and this could lead to that this change in susceptibility is one factor for why we might see this variation in the relationship between biodiversity and disease in nature and also across studies. So with that, in this study, we asked how do pollutants influence the biodiversity disease relationship by looking at manipulative studies in the lab. One limitation of these types of studies is that they're highly controlled and it doesn't reflect a lot of the complexity that we find in nature. So in addition to these lab studies, what we hope to do in the future is to also ask, can we better understand how pollutants influence biodiversity disease relationships in the field? In the field, of course, one major limitation and one challenge for scientists is that it is highly labor intensive. And so what we hope to do is to use the community to help us collect data. And in helping us to collect data, one thing that is important is to ask, how do we ensure that we can properly communicate our research to ensure that the quality of data collection are high? To address this, as I mentioned before, I will talk a little bit more about how we are using art and science to improve public understanding of wetland concepts so that we can convey materials to improve the quality of data collected. So we asked, can art improve public understanding of wetland concepts? To understand this, we focused on third graders as our model group. We chose third graders because recent studies suggest that children at this age are excited about nature, and teaching them about nature at this age maintains their excitement into adulthood. So to help third graders understand wetland concepts using art, we wanted to teach third graders about trematodes as a test. So what we did was a 3-step process. In step 1, we used art to develop content to teach third graders about parasites. These are children in our local schools. What we did was we generated a children's book. This is the development of the children's book. This is the original story. Our artist, Ben McLaughlin, translated this story into a storyboard. And this is where the children's book looks like today. Gemma Jones visits Nuthatch Hollow. And Gemma Jones in this story meets the parasite, the Cercaria, and she follows the Cercaria through the complex life cycle. And we're using this book paired with a lesson plan to teach children about the parasite life cycle. We then take this material to teach the teachers, the children's teachers. We train the teachers how to use this material, and the teachers will bring their students on a field trip to a local museum where they will interact with the members of the community that they read in the book. And so thus far, we've reached 593 third graders. And what we did was we asked them to fill out a survey, a quiz before and after participating in this event. So what I'm showing here is just part of the quiz. On the x-axis, I focused on the parasite question. We wanted to know, again, whether the use of art in the form of a children's book could help teach children about parasites. We asked them to identify a parasite. We asked them if parasites are bad for the ecosystem, or are parasites important. That's the x-axis. The y-axis is proportion correct. And what you can see is in all cases, after participating in the lesson, students improved in their ability to answer these parasite questions. And so where we are going now, as I mentioned before, we would like to use art as a tool to train community scientists on how to collect data on do pollutants influence the biodiversity disease relationship. And so our previous work suggests that the children's book, the art in the form of a children's book, was an effective way of communicating science. And so what we hope to do now is to follow up on that work. We have generated a graphic novel called Amphibian Adventures that will teach children about the biodiversity of amphibians in the wetlands. This was generated by artists in my lab. So in addition to the graphic novel, we've also generated a traditional lesson. This lesson is just a traditional PowerPoint. And so we have these two resources that we use to train teachers to use to teach their students. And then what the teachers will do is that they will break up into the two groups. Half will give the graphic novel lesson, half will give the traditional lesson to their students. So they'll teach the third graders. And then the third graders will be tested on how the graphic novel or how the traditional lesson affected their perception of science, as well as their understanding of biodiversity of amphibians and disease. From here, after testing them and understanding how the art influences their understanding, we will rely on the third graders to help us collect biodiversity and disease data. Again, the goal is to use art as an effective tool for communicating research to third graders to train them to help us collect data on biodiversity and disease. Eventually, we'll have data that looks like this. This is the local pond, and each class will collect amphibian biodiversity from each location. They'll help us graph biodiversity, and then we'll sample disease. And so in the end, we'll look at how pollutants influence this relationship between biodiversity and disease in the field. So with that, I'll transition to the final part of this talk. So far, I've presented some work on secondary salinization on simple host-parasite interactions. We talked about how salt can modify the effect of host-parasite interactions in multi-host communities. And then I highlighted how we're going to use art to help communicate science to enhance community science initiatives. And in this last section, I'll talk about how changes in abiotic conditions may exacerbate or dampen the effects of salt on hosts and their susceptibility to disease. So again, the broad question that I'll ask here is, how do shifts in abiotic conditions modify the effect of sodium chloride? One way that abiotic conditions can change from location to location might be due to the input of leaf litter into aquatic ecosystems. Leaves from various plants contain leaf chemistries that differ, and when they enter into aquatic ecosystems and degrade, this can also change abiotic conditions of the water, so water quality. And so a major threat to wetland ecosystems are invasive species. So we're interested in understanding, as communities shift from native plants to invasive plants, and invasive plants are depositing their leaf litter into these aquatic ecosystems, how do these shifts in abiotic conditions caused by invasive plant leaf litter affect susceptibility to salt? And then secondly, does invasive leaf litter modify host-parasite interactions? This is work done by Dr. Devin DeJacopo in collaboration with Dr. George Meindl. This is work that is done in the classroom. So students enrolled in a plant biology class participated in designing the study, carrying out the study, and also writing up the study. So to address these two questions, what students did was they went out and collected leaf litter from these 6 different species. In white outline, these are the native species. In red, these are the invasive species. We paired the native invasive species by where they're found in the wetland. After collecting the leaf litter, we soaked the leaf litter in water for 28 days, strained out the leaf litter, and placed the leaf litter leachates in these beakers. In each of these beakers, there's also a single embryo of either a leopard frog, which is native to our area, or the African clawed frog, which is non-native in our area and also invasive in some parts of the United States. And so again, the question that we're asking here is, how does living in different abiotic conditions caused by invasive leaf litter leachates influence tadpole susceptibility to salt? So once these embryos reached the tadpole stage, we conducted a standard toxicological assay to see how susceptible they were to salt. On this figure, on the x-axis are the plant species, either native or invasive. On the y-axis is average time to death when exposed to salt. For the plant species, I combined, for the sake of time, native versus invasive species, because we found that native leaf litter chemical profiles were more similar to each other than invasive leaf litter leachates. And what did we find? We found that for leopard frogs, those reared in invasive plant litter were less tolerant to sodium chloride. So something about the invasive leaf litter leachate made the leopard frogs more susceptible to salt. In contrast, we found the opposite pattern for the African clawed frog. African clawed frogs reared in invasive plant litter were more tolerant to sodium chloride. So this is really interesting to us because it was consistent with the idea of invasive facilitation, where invasive plant species seem to facilitate the non-native African clawed frog, but harm the native leopard frog. And so we were interested in trying to understand a little bit more about what might be driving these patterns. And so the next iteration of students in the class decided to ask this question. Why does invasive leaf litter make leopard frogs more susceptible to sodium chloride, but African clawed frogs less susceptible? They conducted a number of analyses on the leaf litter leachates. For today, I'll focus on just general hardness, which is the combination of calcium magnesium. I chose general hardness because we know that invasive leaf litter leachates have a higher general hardness compared to native leaf litter leachates. And importantly, we know that leopard frogs have a general hardness preference that is lower than African clawed frogs. So we hypothesized that leopard frogs, when reared in invasive leaf litter leachates, were more susceptible to salt because the conditions in invasive leaf litter leachates fell outside of their general hardness preference. Whereas for African clawed frogs, the invasive leaf litter leachates were within their preference. So to test this, we repeated the study, but this time instead of generating leaf litter leachates, we just manipulated general hardness, which is the figure on your right. The figure on your left is the data I previously showed. So what you can see is that the data are consistent after manipulating just general hardness, suggesting that the reason that African clawed frogs did better in the presence of invasive leaf litter when exposed to salt, where leopard frogs did worse, might be somewhat related in some cases to general hardness. So with that, the main take home here is, again, the toxicity of sodium chloride is influenced by shift in abiotic conditions. This underscores the importance of taking an ecosystem level perspective when trying to understand how pollutants influence organisms. Now, lastly, does invasive leaf litter modify host-parasite interactions? So along the theme of today's talk, we know that the presence of invasive leaf litter leachates modifies host susceptibility, tadpole susceptibility to salt. How does this change their susceptibility to disease? In this study, Devin exposed American toads to native and invasive leaf litter leachates, moved them to clean water, added parasites, and counted how many successfully encysted. It's important to note that we did the exposure to parasites in clean water here so that we could isolate the effects of leachates on the tadpole without confounding the effect of the leachates on the parasite. We also did the reverse experiment where we put the trematodes in the leachates, but for the sake of time, I'll just focus on this today. What Devin found is that for both native invasive pairs, being raised in invasive leaf litter leachates made tadpoles more susceptible to parasites. So again, highlighting the importance of taking into consideration abiotic conditions, an ecosystem level perspective when trying to understand what influences host parasite interactions. So with that, brings me to the end of this talk. I highlighted how we have taken a tiered approach from the individual to the multi-host community to the abiotic ecosystem level. And the last thing I wanted to leave you all with is that I just spent about 30 minutes talking about a single pollutant, secondary salinization, sodium chloride. In nature, amphibian populations are going to be exposed to a wide diversity of different pollutants. And so one of the limitations of this tiered approach is that it takes many years to build up in complexity. And so what we're also hoping to do is to simplify a little bit to understand generalizability. Are the effects of pollutants on host parasite interactions generalizable? To do this, we've leveraged the incredible students at Binghamton University by having them choose a pollutant of interest and conducting a very simple study. What does this pollutant, their pollutant of interest, do to simple single host parasite interactions? And so these students have conducted this study, many of which are published, and they have found that these different pollutants do very different things to susceptibility to parasites. So where we hope to go next is, can we make predictions about when different pollutant stressors are likely to lead to increased susceptibility to parasites? We hope to do this using machine learning or modeling approaches. So with that, to sum, we are the Qual Lab, and we're interested in the effects of pollutants in the environment and how they influence host-parasite interactions. From the host perspective, we apply a tiered approach. We look at how pollutants affect individuals, host-parasite interactions with multi-host communities, and also integrate in that ecosystem level perspective. Additionally, it's important for us to find ways of communicating our science to broader audiences. Specifically, we've used art as a way to help train community scientists to help us collect data on the biodiversity disease relationship. So with that, there are many funding agencies to thank that have helped fund this project. And importantly, these are the students in my lab that have played an incredible role in collecting these data. This work could not be done without them. And lastly, I wanted to note that we are currently at Binghamton University, but are transitioning to the University of Wisconsin in Madison. The lab will be recruiting a student or a PhD student and a postdoc soon. So I wanted to leave this slide in here in case anybody might be interested or know of any students or postdocs that are looking for positions. For more details, please scan the QR code that is on this screen. So with that, if there are any questions, please feel free to reach out on Twitter, on Instagram, or even via my email, which is jhua@binghamton.edu. Thank you so much for your attention, and I look forward to hearing questions if there are any.